I'm going to be talking about this implementation study. It was an 8-month study. Primarily, the work was done between CATH and Swiss Model. Also, other groups, PDBe and InterPro, are involved as well. So, as an overview of what we're going to discuss today, first of all, some background about what sort of scientific goals we're trying to achieve, and then some of the actual implementation details in terms of decisions like this technology stacks, interfaces, development, deployment, benchmarking. It's kind of this is building APIs, sending data, and checking everything is making sense. There will be a live demo to show that it all kind of works, and then with some points after that. So just to kind of give the original aims of this project, it was to increase interoperability between 4 ELIXIR resources, which are CATH, Swiss Model, InterPro, and PDBe, by building APIs that facilitate the import and export data between them. And ideally, this is to improve the provision of 3D models for protein domain sequences via CATH, Swiss Model, and InterPro. So the resources involved are listed there. I'm going to give more detail about each of those in a bit. So just to give a tiny bit of background, if you've never come across the idea of homology modeling before, the idea is that you take a protein sequence, you look for other structures that we already know that have a close similarity in terms of there's a homologous match between your query sequence and the template structure, and then based on the alignment between your query sequence and that template structure, you can build a 3D structure. So really, that's the kind of the overview of the pipeline. In terms of how we are kind of putting that together with the pipeline that we've done here. Again, we start with a query protein sequence. We go off into the CATH API to get some information about which related template structures might be very useful to do that modeling step. And then we pass that information over onto Swiss Model via APIs to go off and actually do this modeling to get the 3D structure. After that, we're pushing this 3D data, these kind of 3D models, predictions into a resource called Genome 3D, which is also part of this collaboration. And that resource holds consensus information from lots of different prediction groups in terms of predicted structures from sequence. And then from there, from Genome 3D, that data is also further disseminated to other resources such as InterPro and PDBe from the EBI. Just a brief word about CATH. CATH is a protein structure classification database. Essentially, we take structures from the PDB, we chop them up into structural domains, and we classify them according to their evolutionary superfamilies. So essentially, you can already search a sequence against CATH and find matches to what we call FunFams. I'll mention what they are briefly. So the FunFams are an important part of this grant, so I'll try and give an overview of this without going into detail. The idea is that CATH is a classification of structural domains from the PDB. On top of that, we add in lots of predicted structural domains from UniProt, so we use sequence searching technologies like lots of domains that we think are going to be a certain structure, and we group those together into a superfamily. And this kind of joint resource is CATH Genome 3D, which is structures and sequences together. If we were to, rather than colour them by whether they have a structure or not, if we were to colour them by whether they have a particular function, then this is kind of the starting point for the FunFams, what we're trying to do with these functional families. And the first step of that is to separate all of these individual sequences within a superfamily into the type of function that they perform. And each of these will have a unique sequence fingerprint. What we know from our own research is that within these functional families, because function is highly constrained, we also see that structure is very highly constrained, which gave us a good indication that the structures, if we find a good match to a functional family, then we should also have similar structures, which is where we came into this the pipeline of modeling. However, the last couple of steps about aligning and generating a 3D model, that's already handled incredibly well by groups such as Swiss Model. So at this point, I am going to, so the idea is that we kind of select the structure representatives for templates, and then we pass that information on to the experts in modeling, which is Swiss Model. And speaking of passing over, I'm now going to hand it over to, where are we? Sorry, I'd like to hand over to our Swiss Model team, but there are so many people here that's tricky. Oh, you got it. So Gerardo.
Yes, okay, thanks, Ian. So let me give a quick introduction in what Swiss Model does. So Swiss Model is a web server to predict 3D models from protein sequence. So as an input, you give the sequence of the protein, and then you can run a fully automated pipeline that ends up with 3D coordinates of the predicted three-dimensional structure for that protein. So the main intermediate result of that, as was mentioned also earlier, is finding proteins which are homologous to the target sequence and which have a known structure and which can be aligned to the target sequence. So this is what we would display as template results. You get a list of these template structures with their alignments. We rank them very carefully and try to select the ones which we think are best suited for modeling. And it's exactly this step where now in this collaboration, we want to feed in information from the FunFams to give us better templates, better input for the modeling engine. Now the last step, once we have this alignment to the template structure, we feed this into our modeling engine and we display the modeling results, which are the 3D coordinates fully atomistic of the target protein, plus our own estimates of how good we think the model is, how reliable different parts of the models are, which here you can see that in blue and orange being bad residues, blue being good residues. So our contribution here from a technical perspective, what we added to our pipeline is that we put in an API where you can, via scripting, feed in the alignments into Swiss Model, provided by using the FunFam APIs, and then we produce the final 3D coordinates together with our model quality estimation, which is the final result of the pipeline. And then I hand back to Ian that will tell you more about Genomes 3D.
Lovely, thank you. So that was kind of covering the first bit of the pipeline, and then talking about where the data goes afterwards, which is another important part. Is it first goes into this kind of repository of structural predictions called Genome 3D, and the idea is that this is an organisation that already existed. That was lots of different structural prediction groups, such as Few from Tom Rundell, Fire from Mike Sternberg, David Jones, and so on, providing structural predictions of uniprot sequences. And the idea being that if you look at all these different structural predictions lined up in a consensus approach, you can see where these prediction methods agree and where they disagree, and that can help to give you an idea of which regions of your predicted structure are more likely to be reliable. So this resource already has been around for a while, and groups are adding annotations. So sites like InterPro, we've been developing ways of allowing InterPro to bring data, this kind of predictive structural data, across from Genome 3D into InterPro, which actually has been done. I can show you the results of that. And the other partner in this collaboration was the PDBe, and again, this is about bringing structural data into other resources from Genome 3D. So in terms of the implementation, these are the kind of the basic points that I'm going to be talking about. So we already had CATH and Swiss Model as existing resources with their own services and APIs and so on. Some of the pipeline that we've discussed so far wasn't already implemented as an automated API within the CATH resource. So we started to build code within fresh code on GitHub as CATHSM server. So this CATHSM server was the kind of the first API point where we have a query sequence and we get our potential structural templates back from that. We're using the code within CATHSM server. CATHSM is CATH Swiss Model. We're using code there to essentially mediate the API endpoint that was already in existence in CATH. We didn't want to make too many changes to that because people's code already already depends on that not changing too much. So all of the new code for this project is in GitHub as CATHSM server. There is another project, CATHSM Clients, which is the command line tools and libraries which talks to the server to go off and make these sort of mediate these API requests. And the second API request was, once you've got these templates, can you send all that information off to Swiss Model to get a really nice 3D structure. So in terms of how we went about arranging this, again, it started off as an 8-month project because we were all working on other projects as well. It extended to more than 8 months, but we started by kind of agreeing what tools we're going to use, which essentially for wiki documentation, issues, and code. We use GitHub. We created our own organization to kind of keep things tidy. We've got automated builds, so the code is continually being tested, unit tested as part of that build to make sure we know when things break. Also, part of the testing, we kind of need to make sure that the tests that we do have are actually testing all of our live code, which is where the code coverage comes in. So that's just kind of giving us an indication that the tests that we are using really do cover as much of our live code as possible. And then the other thing to mention was because we've got lots of we're kind of interchanging code and we're interchanging data between groups and resources within groups. So having kind of stable releases gives you a fixed target to work on. All this code is available to use. In terms of the basic technology stack, this is something that we kind of settled on pretty early. We try to use data standards where they looked appropriate, including OpenAPI, which is kind of platform independent way of specifying RESTful APIs. Also, each of the individual backend APIs requires login accounts and requires username and passwords. So we use kind of standard protocols for authorization, which is OpenAuth. And then in terms of the technology stack for building the client and the server, Python 3, Django, REST framework, and there are some kind of dependencies that take time to run. So if we need to run something asynchronously, then we're using Celery as a queue with offline workers to basically get jobs done outside of the web environment. And on top of that, relational database and so on. One of the very first sort of things that we designed was to try and make sure that the original CATH API and the Swiss Model API, shared kind of a general architecture. So a lot of these jobs are asynchronous. So the very first thing you need to do is we get authorization to say that I'm allowed to submit, and this is me. I'm allowed to submit, which essentially is a post request to API token auth. That gives you a token ID that you can use for the subsequent operations, which is essentially to submit some data, which is a query protein sequence, to a given operation, which is go off and select me some templates. After the submit, you get a unique ID back, which you can continually check until it's finished, in which case you can get the results. So this kind of is the general architecture for asynchronous jobs. I'm sure it's shared by many other kind of implementations. The nice thing about the Django REST framework, if you haven't already used it, is that you kind of provide the code to define what the API is, and it takes care of all of the specification and the documentation. So it kind of generates lots of nice documentation directly from the code, so you don't have to kind of maintain that, which is very useful. So at this point, I am going to hand back over to the Swiss Model team to talk about deployment. Can I do this? Oh, it's done.
Oh. So, yeah, hello, this is Stefan from Basel. Yeah, I have to tell you something how we deploy the thing. Ian already mentioned that we use Django, which naturally means we have a database in the backend. We also have Redis for caching or to talk to Celery. But instead of going for traditional setup with bare metal or virtual machines and installing everything there, we thought we try containers in this case and put every of the third-party services in their own container, simply because it makes it easy to maintain the software, to set it up, because you start with a base image, for example, for Redis. You can get this from Docker Hub. all you have to do is configure it, and that's it. Yeah, and to run all of this, we use Docker Compose, which gives us a single command solution to build all the containers or start them, restart them, and so on. So this is a basic workflow, basic setup how this works. On the right, you have the client externally talking, in our case, to Nginx, which gets its data to provide to the client from our deployment, in that case, Docker Compose. If you want to boot this deployment, if you want to start it, you usually use a file called Docker Compose YAML. That's just the definition of all the services you need and which all the services work together. And in case you do not have your containers or your images already locally in your environment, Docker Compose can also build them for you. This requires the definition files for the containers. In terms of Docker, that's the Docker files, and they fetch the base images from, so the basics for the containers from Docker, from a container registry. In our case, it's Docker Hub, and our own project code is provided via Git. And now I quickly want to walk you through our Docker Compose YAML file, because during the project we noticed everybody has an idea about containers. Not so many people actually set up their own containers or a complex container, and even less people have deployed something with Docker Compose. So on the left hand, you see just where in our container landscape we are, and on the right there is the compose.yaml file. And they usually start with a version declaration, because Docker Compose is a bit picky about the versions and incompatibilities in between this. Below this, we define volumes. That is because the containers are stateless. That means if you restart a container, it lost all the data you put into it since the last time you started it. So we need storage for database, it's DB store. We need something to keep the static files for the internet, for the web page, and something to store the log files of Nginx in. And below that starts the service section, where all the containers in the end we want to run are defined. And we start our definition with Redis, which just has a restart policy set to always. That means whenever this container is down for whatever reason, Docker Compose will restart it. And that's the default for all of our other containers. Below the restart policy, you see build instructions. That is, in this case, pretty simple. We tell Docker Compose just check the Redis subdirectory in your directory tree, and you will find the Docker file there and a config for Redis, and that's all how you declare how to build a container to Docker Compose. Below this, there is a special thing for Redis. We had to define an additional host name, that's CATHSM-API Redis, because just while working with containers, suddenly they couldn't see the Redis container anymore. Our second service we have is actually the Django container for the CATH API, which has something special for the build section. So what we wanted to do there is, well, traditionally, inside your Docker file, you would instruct Docker to always do a fresh checkout of a Git repository if the code is based on Git. But we also wanted to enable us to build the containers from local changes we did to our local checkout. So you develop something new, you want to test this. This is possible with this, so you just do Docker Compose build, and it will rebuild your containers, and you can test your latest developments. This is controlled by the args section in this build block. Below this is just the definition on what Django depends. Of course, we need Redis and we need Postgres as the database. Otherwise, Django doesn't run. And in that way, Docker Compose makes sure that Django is only started after Redis and Postgres, and is also shut down before Redis and Postgres if you want to shut this down. And below is a definition of environment variables for to be seen inside the container during runtime. This can be done, for example, with an environment file you provide, and then you can use variables to declare your environment variables. And below that, in the volume section, we attach our static volume to the mount point /static inside the container, so that Django, when it collects the static files from its own project, can provide Nginx. And the last thing is a command which is executed once the container is started. So the Postgres setup is actually simpler than the Django one. The build section also has something special there. That's an SHM size declaration that comes straight from the Postgres documentation, which says Docker usually runs with not enough shared memory. Please increase it when you want to run it. In the environment section, the special bit is that we tell Postgres to run with the SHA-256 checksums for passwords. Yeah, and as volumes, of course, we attach the database storage volume to the to a mount point where Postgres would usually write the data to. That means that now, if the Postgres container goes down and up again, the data still exists. And this entry point definition you see there is written in that way so that we can pass parameters to the Docker endpoint script. If you write it in a different way, then the endpoint script would assume that everything which comes after it is actually some command to execute after the entry point script has run. Then the Nginx declaration that really only uses the base image from Docker Hub. And in the build context, we have a dedicated configuration file for Nginx, and that's all that's special about this. As volumes, of course, we attach the static directory, which we already attached to the Django container, so that when Django puts the static files in there, Nginx can see them below /static. And we also mount in the volume to keep the log files. And in the end, you have to declare ports with which you can talk to the container, to the Nginx, to the web interface. And the last container is the Celery, that's our worker. It has the same build instructions as the Django container, so that if you change something in the worker code, you can test it without first uploading everything to the Git repository and then downloading it again. Yeah, and there the command you see is basically how we start Celery. And that's it for the deployment. I think what we have now is Xavier with benchmarking.
Yes, so now that we have this fully running setup that's running fully automated, we need to make sure that everything is working together properly. And also because the goal is to send data in the end to Genomes 3D, we want to ensure that we are really sending the best possible models that we can do. More specifically, we want to compare how the new CATH Swiss models differ and how the implementation differs from the automated Swiss model pipeline that we know, and how accurate the predicted models are. So the question naturally that we're asking is what's a good data set for that? And we actually in-house also have the Cameo, the continuous automated modeling evaluation server, which is running weekly and sending 20 new 3D prediction challenges each week. It basically hinges on the PDBe pre-release of the amino acid sequences, which is part of the PDBe weekly release cycle of macromolecular structures. On such a day, the PDBe pre-releases the sequences of amino acids and the ligands that are part of the structures that will be released on Wednesday. So what Cameo does is that it gathers these sequences, chooses 20 targets, and send them to the participating 3D modeling servers. And these servers have up to Wednesday at midnight to return their predicted 3D structure models to Cameo. And at that time, PDBe releases the actual experimental structures, and we can then assess, evaluate, and score the predictions. So in this specific case, we had to make a few changes to the standard Cameo pipeline, in particular because Cameo focuses on full target sequences, and we're actually interested in domains. And Cameo basically penalizes short models and really wants participants to send models with a larger coverage, but we're not interested in that in this case. So we modified the scoring scheme a bit to score domains instead of the full target sequence, and that basically means cutting the target to the domain. So what do we expect from the two pipelines? So the CATH Swiss model pipeline now defines the domain of interest in this benchmarking experiment. And we also expect it to maybe find better templates, especially in case of remote homology. However, we know that the alignments might not be optimal. On the Swiss model part, we have more recent templates. We have a weekly update cycles for our templates. And we also participate in Cameo and have an optimized logic to select the best template together with their alignments. However, the target sequence that we had in this case was the full sequence, and so the model for the domain may not be fully optimal. So what we get with this API, so we send to the API 1 the full target sequence, and the API 1 and FunFams finds hit on this target sequence. And so in this case, we had 7 hits and generally 2 domains that show up. The next step is to use the CATH resolve hit script, which collapses the list of domain matches in the query sequence down to the best non-overlapping subset of hits. That is basically the domain architecture. So in the case of this target sequence, we'll have 2 domains in the benchmark, and so we compare that with the Swiss model the model generated by the Swiss model fully automated pipeline, which got the full target sequence and generated a model. In this case, with the 2 domains, it could happen that we generate 2 models for the target sequence. So here we would simply cut the model along with the domains, and we do the same with the target reference models. And so in the end, we found matches. So we took Cameo target from August 2018 to January 2019, and we found 205 targets with hits in FunFams. And this yielded 760 hits. So domain template alignments with API 1, and with API 2, we were able to generate 760 models. 247 of them were resolved in continuous domain architecture, as I explained before. And we compared that to the 369 models that Swiss model had sent to Cameo for the corresponding targets. For the score, we used LDDT, the local distance difference test, which looks at the conservation of atom contact in the reference and in the predicted structure. It's superposition free, and in this case, we cut on the domain to really look at the domain itself. So in the end, we plotted the CATHSM and the automated Swiss model results, and so for Swiss model we chose the model that was performing the best on the domain based on the average local Q-Indisco score of the domain, so based on our high quality score. And the score itself that you're seeing on the plot is LDDT, and as expected, the 2 pipelines perform mostly fairly similarly. Most of the points are on the diagonal. There are a few outliers, of course, where the two pipelines performed a bit differently. So we are investigating this. So in conclusion, we demonstrated the functionality of the pipeline. We have robust API, they work, and they produce high-quality models. And so this benchmarking exercise also helps us to learn from the differences. In particular, we realized there's a bit more work needed to really understand the differences. This is, of course, beyond the scope of this implementation study, but it could lead us to better combine CATH and Swiss model. And in particular, we already identified that it should be possible to refine the alignments to the templates. And with that, I will give the floor back to Ian for the demo.
Great, thank you. Yeah, so from a like a sort of generic point of view, we thought that the design of the data set was really important, just so we can always find out when we're making changes to the pipeline, we know whether those changes are going to have an improved results at the end of it. So you know, not just as specifics, but just like as a general idea of like why we considered the data set very early on and right the way through. It just kind of gave us a really good handle on whether the changes we were making would actually improve the final result, which was an important part of the work. So now the bit everyone's waiting for secretly is to see a demo that goes horribly right. Let me just share my screen. Right, so I'm assuming that you can see my screen now. So I am going to, so the CATHSM server is on, I need to get access to my toolbar, it's here. So we have an implementation of the CATHSM server, which is already been, it's already live, it's not here, it is here. So there's a very simple front end, just with some basic information about the project and about how to access it. If I click on this one, it takes me to the CATHSM client, which gives information very, very briefly. If you're not aware of Travis CI, this is kind of effectively what it looks like. It takes it takes every single commit that you make to GitHub and then runs all of your tests in a very kind of independent environment. And at the end of it, if your tests, when I get to the end of it, if your tests pass, then you get a nice green tick. And again, this just gives you a really good idea without making any effort whatsoever. You know that everything is currently working. And also code coverage, just to kind of give you an idea what that is doing. If when you run the tests, what that is basically doing is checking all of your live code to see that when you run the tests, it checks which bits of your code are actually being tested during that test. So this is the live code, and the green ticks are basically saying that these are the bits of the code that gets tested during the tests. The red bits of the code are things that don't get tested. So for example, in this case, the tests I put together do not check whether this is true, and therefore that is a statement that is not officially tested by our unit tests. So it's just kind of a useful indication that what we think is being tested is actually tested. Anyhow, let's go through to actually getting this installed. So if I take a clone of that and get into that directory, we always recommend doing work in a virtual environment. We're using Python 3, so once that's done, we can activate our virtual environments and making sure that all of our kind of local environment is tidy and kept separate. So this is me just installing the dependencies. Again, we've tried to make sure that these dependencies are reliably installable and are clearly versioned to try and make sure that everybody's installation is the same. And again, the automated builds help with this. because if it doesn't work for the automated build, then it probably won't work for everyone else as well. So now I've got the dependencies installed, and I should just be able to have access to one of the scripts. So this is the script. If I show the usage, this kind of gives you effectively access to the pipeline. So if I use there are some example data within this. Let's have this one. So this is just a simple protein sequence in a FASTA format. So if I were to take this, I can say what the input is going to be, which is this test. We need to give it an output. I will say results directory, and then this is the bit that I will refer to the user credentials that we've got access to in GitHub. So I'm showing you passwords, but these passwords are just the same passwords that are used by the test suite. So they're not that secret. So once you have the usernames and passwords that you can use, this hasn't asked me for my password because I've already inputted that username and password, and it's stored a local token on my computer from previous runs. So this, what you're seeing now, is the query sequence that is being searched against, has been thrown over to the CATHSM server. These were the results, all of the results, and then it goes and takes the best ones and sends them over to SwissModel. This is the alignment that it sends over to SwissModel. SwissModel has come back because the results are cached. It didn't take very long to say that it's completed, and we have access to that here. And if I open up a right, so we can actually access that. So then we can kind of see the protein structure that's been put from the original protein sequence at the end of that. So if I can stop sharing my screen, right, and go back to the demo. So what we're seeing there is the pipeline working. Obviously, people can, humans can use it in that way, but really that is set up for these kind of automated pipelines to happen. So it's not just the user interfaces, it's the libraries behind that we hope will be useful for further work. In terms of disseminating this data, I mentioned that this data is going into Genome 3D, which is a repository of structural predictions, and from there it's going on into InterPro and PDBe. And this is the second part of the implementation study. This involved creating new API endpoints within Genome 3D, which was done in collaboration with the InterPro team. The InterPro team have now included these Genome 3D annotations as a result of that work. They've included them, and these are the live InterPro pages, which now have a Genome 3D tab, which show all of these structural predictions. So that we kind of got that end of the pipeline up and running as well. So these are live pages on InterPro. Just to mention also, in terms of PDBe and generally as 3D beacons, that this kind of transfer of structural data, structural prediction data, and the design of APIs and storing the data and so on, this kind of moves into another ELIXIR community, the 3D bioinfo and the 3D beacons work, which is kind of happening in the future. So all of this hopefully leads very nicely into that as well. So just in summary, this was the original breakdown of the project. It lasted for a lot longer than this because essentially, as I mentioned, we were all doing other jobs as well. So I think it's in the end probably more like 16 months, maybe 18 months, maybe rather than 8 months. But it has covered a lot of ground. I think it's been a really successful collaboration, and I think we've all enjoyed it as well. So I think in terms of just acknowledgements from our side, CATH Genome 3D, it's myself and Christina Rengo. The SwissModel team, you've heard from most of them. Also, InterPro and PDBe, so they help in doing the other data dissemination as well.